Genetic disorder that causes tumors to grow on my body. Would you like to learn more about it?
I don't have a lot of neurofibromas or a lot of capillary spots that's visible to the world.
Just look, you can't see who I am. I guess that's the answer to the question. You can only see what you can see, which is true of anybody.
Make NF visible for those who don't have a visible aspect. They have brain surgeries. I've had brain surgeries. Sometimes when people don't understand what something is, they get scared.
It's hard.
It's really hard when you're young. Then you really have to deal with that, and you have to deal with constantly being stared at, reminded that you do look differently.
When you're faced with a disorder like NF, and if it runs in your family, in my particular case, my son has it, that's how we found it.
Not a lot of people know that I had 5 surgeries.
Even just doing simple tasks sometimes take a lot more out of me because my body is fighting this.
I'm not freaky, I'm not weird, I'm not some alien. I'm like my other peers, I'm like everyone else.
Sometimes I get scared when they see my tumors.
When I walk into a room, I feel really shy.
I wonder whether or not I should tell them or not, or what, and if I do, are they going to see me differently? Are they going to understand?
I feel like it's kind of coming for me eventually, just having these tumors.
When I'm experiencing pain or symptoms because of my NF, I feel sort of guilty.
I have one of the more unique things with neurofibromatosis, which is called pseudoarthrosis.
It takes more of a mental impact on me.
Someone might ask me why I have so many birthmarks, and I tell them that it's because I have NF.
It's not so easy to be me.
I fight NF by staying strong even through the hard times and just knowing that everything's going to be okay.
My name is Dane, and I want you to see me.
See me, and see NF.
And see NF.
Good morning, everyone, and welcome to the session on pain in NF. I'm Stacy Martin. I'm a psychologist from the National Institutes of Health, and my co-chair, Rajesh Khanna from the University of Arizona, had a last-minute family emergency and won't be able to join us today. So I'm going to fly solo here, and we're going to get right into the talks. The first talk is by Dr. Taryn Allen. She's a pediatric psychologist at the Frederick National Laboratory for Cancer Research, where she supports research in the pediatric oncology branch at the National Cancer Institute. And her presentation is on the impact of a psychological intervention on heart rate variability in adolescents and adults with NF1. So we'll have Dr. Allen start now.
Hi, my name is Taryn Allen. I'm a psychologist at the Frederick National Laboratory for Cancer Research, which is sponsored by the National Cancer Institute. And the title of my talk today is Acceptance and Commitment Therapy and Heart Rate Variability, Implications for Pain Management in NF1. I don't have any disclosures. I just want to start by giving a little bit of background on pain in NF1. Increasingly, we are really understanding how common pain is in this population. A recent report from 2018 suggested that about 70% of adults with NF1 experience pain, and while pain can be linked directly to plexiform neurofibromas, it can also be related to numerous other NF morbidities such as headaches, GI distress, or scoliosis, among other things. Chronic pain in individuals with NF1 has been associated with reductions of quality of life and worse socio-emotional functioning. And unfortunately, for many individuals with NF1, pain relief can be very difficult to achieve. While we know quite a bit about the impact of pain on quality of life in this population, we know far less about the impact of pain on physiological parameters and really have a limited understanding of the mind-body influence of pain in this population. We know that chronic pain has a pervasive impact on how the body functions, and we've also learned that the autonomic nervous system plays an important role in the emergence and persistence of pain in other populations. Heart rate variability is a specific measure of autonomic health, and on a technical level, it reflects the time elapsed between consecutive heartbeats. Here on this slide, I have a tachogram, which is essentially a graph depicting heart rate variability. The y-axis shows the time that's elapsed between consecutive heartbeats. So on the left-hand side, we have an image of high heart rate variability where you see high peaks and low peaks. And really the autonomic nervous system here is responding flexibly and is adapting to different experiences this person is having. This is in contrast to the image on the right-hand side where we see low heart rate variability, which is essentially like a flat line, and you're not seeing that adaptability or that responsiveness to different experiences in the everyday environment. Greater heart rate variability has been associated with better overall health and emotional well-being. The saying goes, a variable heart is a healthy heart. And in contrast, lower heart rate variability is observed in individuals with chronic pain and psychological stress. We had an opportunity to look at some data on heart rate variability among individuals with NF1. And our research suggested that the ability to adapt to pain, which we call psychological flexibility, mediated the relationship between heart rate variability and pain interference in individuals with NF1. Another way of putting this, or to put it more simply, we observed that heart rate variability was influencing specific psychological processes, and in turn, these processes influenced the extent to which pain was getting in the way with everyday activities. This was really interesting to us and made us wonder whether some of the behavioral pain interventions that we use, whether they could have an impact on heart rate variability, similarly to their impact on psychological flexibility and pain interference. Specifically, our group at NCI has been very interested in acceptance and commitment therapy, which is a type of intervention that's commonly used for the treatment of pain. ACT is an intervention that incorporates many different elements. A few core elements include mindfulness or present moment awareness, this idea of acceptance, which is really acknowledging that pain is a part of life. So rather than fighting or trying to avoid or trying to get rid of, rather than putting our efforts towards that, can we say, okay, pain is here, how do I work with it? And then values-based action is another core element of ACT, which is really committing to doing things that are important to you, even if pain is coming up. So pain may be there, but can you still get on with the things that are most important to you? Through these different elements, ACT targets psychological flexibility, or our ability to mentally adapt to our experiences, to mentally adapt in the face of pain, adapt how we confront pain, how we think about pain, how we handle pain. ACT is the only intervention with strong research support for the treatment of pain, according to the American Psychological Association. And just recently, at the end of 2020, the World Health Organization designated ACT as having moderate certainty, which is a fairly high standard for the treatment of pain, and they recommended it as a primary treatment for chronic pain. So with that, I want to get into the current objective of our study, or of this sub-study, which was to examine the impact of acceptance and commitment therapy on heart rate variability in individuals with NF1. We also wanted to further examine the relationship between pain and heart rate variability in this population, which is an extension of our research from a few years back. So just to get into our methods, participants in this study included individuals with NF1 between the ages of 16 and 64 years old who had at least one plexiform neurofibroma and chronic pain for at least 3 months. Everyone who participated or who was included in the data analysis that I present today participated in a larger RCT of ACT, which aimed to improve pain interference. The ACT trial included a waitlist control group, and an immediate ACT intervention group. It should be noted that everyone who participated in the waitlist control subsequently enrolled in the ACT intervention phase of the trial. All participants completed baseline questionnaires that assessed their pain and psychological well-being immediately prior to the intervention. This included the NRS 11 for pain intensity. We looked at the PROMIS pain interference scales. We also looked at psychological inflexibility using the PIPS. We looked at depression and also pain anxiety. Everyone also completed a 5-minute EKG from which a measure of parasympathetically mediated heart rate variability was obtained. These same assessments, the questionnaires and the EKG, were completed again following the ACT intervention for everyone who participated in this study. We looked at correlations to examine the relationship between heart rate variability and pain-related variables at baseline. And then to answer the question, does heart rate variability change following the ACT intervention, we used 2 different types of repeated measures ANCOVA. First, we did a between groups analysis looking at the ACT group compared to that waitlist control group. And then we also looked at everyone pre-ACT to post-ACT, looking at whether there was a change, and this was a within groups analysis. So we did both of those. And then finally, we used a linear regression model to examine whether changes in different pain variables predicted changes in heart rate variability over the course of the ACT intervention. Okay, so let me go through our results. We had 64 participants enroll in our ACT trial. They were about 30 years old on average. Again, it ranged from 16 to 64, and a little over a third of our participants were male. When we looked at baseline correlational data, we found that lower levels of heart rate variability were associated with significantly more pain interference, and lower levels of heart rate variability were associated with significantly more mental inflexibility with pain. So the more inflexible you were, the lower your heart rate variability was. Both of these findings are consistent with the literature in other populations, very much what we would expect. So then to get into our primary question, does heart rate variability change following the ACT intervention? We looked at this 2 ways. So this first table it is presenting data from our between groups analysis, so the ACT group compared to the waitlist control group. And here we don't see a significant difference in groups in their heart rate variability outcomes. Then we looked at, we tried to answer the same question using a within groups analysis. And here we do see a significant change in the post-intervention heart rate variability levels compared to the levels at baseline. So when we look pre-post within groups, we do see a significant change in heart rate variability. So finally, we wanted to ask if pain changes, does heart rate variability also change? We looked at 2 elements of pain. The first was pain intensity, and here we found that that changes in pain intensity over the course of the ACT intervention indeed predicted changes in heart rate variability. So as pain intensity reduced, heart rate variability increased, which was a pretty interesting finding. Surprisingly, however, our pain adaptability, our ability to be flexible with pain, that did not predict changes in heart rate variability over time. And so that was a null finding there. So just to briefly summarize the results, our data do provide additional evidence that heart rate variability shares a significant relationship with pain in NF1. In addition, when we look at the impact of ACT on heart rate variability, We did not see a significant difference following the ACT intervention compared to a control group. However, when we have a larger sample and we look within groups from pre-intervention to post-intervention, we do see a significant improvement in heart rate variability following the ACT intervention. In addition, we observed that changes in pain intensity significantly predicted changes in heart rate variability following the ACT intervention. And finally, in contrast, we did not see that changes in pain flexibility predicted changes in heart rate variability over the course of the ACT intervention. So taken together, the data I've presented today provides some preliminary evidence to suggest that ACT, which is really a low-risk, well-tolerated behavioral pain intervention, may improve physiological markers of pain in NF1, and specifically heart rate variability. We find this really very encouraging because the improvements we're seeing in heart rate variability are in addition to improvements that we observe in pain interference, which we've reported in a manuscript that's currently under review. More research, however, is really needed to explore the physiological benefits of ACT and, potentially other pain interventions in NF1, especially with larger samples. When we look at our data, we do see that the within groups analysis with a sample size of about 60 is suggesting significant improvements in heart rate variability compared to our between groups analysis where we only have about 30 people in each group. When we think about future directions, we'd also really like to see this type of work extended to individuals with other type of NF. We know that folks with NF2 and schwannomatosis also experience significant pain, and that pain interventions and pain therapies have really not provided the extent of relief that is needed for these folks. So we hope that ACT or other novel pain interventions can be considered for a broader population. And here are my references, and then finally I just want to acknowledge our team at NCI. It's such an amazing group, and I'm so grateful to work with everyone there and to have worked on this project with everyone, and also NTAP, who funded this ACT trial.
Thank you, Dr. Allen. That was wonderful. We have a question from the audience asking, besides heart rate variability, did you use a pain diary or additional questionnaires post-intervention?
Yeah, that's a great question. We did not use a pain diary to track pain over the course of the intervention to get kind of that in the moment assessment. We did, in addition to heart rate variability, we did look at pain interference after the intervention, and we saw a significant improvement there. And we saw that both when we look at the ACT group compared to a waitlist group, and also when we look within subjects just pre to post. We also looked at pain intensity, and in pain intensity, which is really, it's an interesting construct because that is not the target of ACT necessarily. It is really, even if pain is still there, can you work with it? Can you do the things that are important to you? And so with pain intensity, we did not see between groups an improvement in that specific measure. Yeah, though I believe within groups, we do see pre-post pain intensity changing and lessening.
Great. And then something I'm curious about is, knowing what you know now and looking back, if you were going to do this study again, is there anything about the ACT intervention that you would change that might kind of increase the impact on heart rate variability? Like any elements of the intervention that you would emphasize more or less?
Yeah, I think that's a really good question. I think when we are considering this construct of heart rate variability, and we're thinking about how the mind and the body are connected, we really think about the kind of frontal lobes of the brain, and that area of the brain is very strongly impacted by mindfulness. And you can see in other populations really a really strong impact from mindfulness on heart rate variability and just overall physiological functioning. So I think in the context of kind of a whole body intervention and emphasizing certain elements in ACT, I might emphasize that a little bit, just because we know that that can have such a direct effect. Yeah.
That's great. Thank you so much for a really interesting talk. Appreciate it. And we're going to go now to Dr. Larry Sherman. He is a professor in the Division of Neuroscience at the Oregon National Primate Research Center and the Department of Cell, Developmental, and Cancer Biology at the Oregon Health and Science University. And his talk is on categories and mechanisms of pain in NF1, NF2, and schwannomatosis. Dr. Sherman?
Hello. Thank you for inviting me to this meeting today. I'd like to acknowledge the people from my lab who did the work that I'll be discussing, as well as my collaborators from my own group. That's Steve Matsumoto, my longtime collaborator, Fatima Benin, Christina Fernandez-Valle, and Sue Eicher. The work that I'll be talking about from my own lab was supported by grants from the Children's Tumor Foundation, the Oregon Health and Science University Neurofibromatosis Grant Fund, a grant from USO Pharma, and the Department of Defense Neurofibromatosis Program. When we talk about pain, there's really two major categories. One is nociceptive pain, which can be from thermal, chemical, or mechanical stimuli, usually not really nice ones, obviously, really resulting in signals being transmitted through the peripheral nervous system, up through the dorsal ganglia, and eventually into the brain where pain is perceived. Neuropathic pain, on the other hand, is caused by damage or disease that affects the nervous system, resulting in maladaptive plasticity of the nociceptive system and contributing to chronic pain. Interestingly, in recent times, it's been shown that Schwann cells may actually contribute to pain. And they do this by releasing pain mediators following injury. For example, neurotrophic factors, prostaglandins, and various cytokines and chemokines can be released by Schwann cells and then taken up by receptors on nearby sensory neurons, leading to pain changes. So the question is, could Schwann cells that are prone to tumorigenesis, as in the NF1, NF2, and schwannomatosis, also release factors that promote pain in these diseases? If we look at the different types of pain in each of these, we see some similarities and some differences. Starting with NF1 patients, it's not really talked about much, but NF1 patients have significant impact, levels of pain that can actually impact the quality of life. The incidence is unclear. It ranges anywhere from 29% in one report to 70% in another. It can be chronic, which lasts for months or even years, and it's typically characterized as painful peripheral sensory neuropathy. The chair of this session, one of the chairs of this session, Rajesh, has released a really nice review article back in 2019 covering this topic, and this is from that review. You can see that NF1 patients have pain in all the different places of their bodies. It's usually related to some sort of tumor, but it could also be related to other things that happen in NF1 patients, things like hydrocephalus, seizures, hemothorax, and scoliosis. So there's a lot of different ways you can get pain in NF1. The question, though, is, do patients with NF1 predispose to pain for some reason, or is their pain, more exquisite when they experience it? And Rajesh's study recently actually addresses this very question. There's a protein called collapsin response mediator protein 2, or CRMP2, and CRMP2 interacts with neurofibromin. So when neurofibromin is lost or mutated, It results in CRMP2 phosphorylation changes and activity changes that in turn influence channels, for example, on neurons that could influence pain signaling. As shown here, that includes sodium and calcium channels that can induce calcitonin gene-related peptide, or CGRP. And CGRP is interesting because its release can influence things like migraines, but other types of pain as well. So this would suggest that just the loss of neurofibromin, not the consequences in terms of the tumor formation, can itself influence pain signaling in patients. Interestingly, patients with NF2 also experience pain. It's really typically associated with schwannomas, suggesting there may be some mechanical relationship. The incidence of this pain is unclear, but it can be significant. In fact, it can impact the quality of life in a number of patients. And painful NF2 schwannomas, interestingly, have been recently reported to have distinct patterns of gene expression compared to non-painful schwannomas from NF2 patients. And in fact, some of these genes may influence pain directly. So one of the genes in this study they looked at was fibroblast growth factor 7, which, as you can see here, is highly elevated in the lot when there's a loss of NF2, but specifically in the schwannomas from these NF2 patients. So there are painful and non-painful tumors, and the painful tumors seem to be different somehow. When you look at FGF7 function, interestingly, in a rodent model of pain, this is using a look at the hind paw stimuli, it looks like FGF7 actually reduces the withdrawal latency, indicating that FGF7 can, in fact, moderate pain responses. So again, compared to NF1, where you had one mechanism, you have a potential other mechanism here in NF2, which may accentuate pain responses in the patients. When we talk about pain and NF, schwannomatosis is really at the top of the list because the patients present with pain often. And this pain can be associated with a mass, or it could be systemic. It's often persisting in these patients despite really aggressive medical or surgical interventions. It can be both nociceptive and neuropathic, and it may differ, interestingly, in patients with different genotypes. Patients with SMARCB1 mutations may have different types or levels of pain compared to patients with LZTR1 mutations. Now, a couple of interesting recent studies have suggested mechanisms for this. One very recent study just came out showing that there are differences in the transcription, which may be regulated by genetic and epigenetic mechanisms in painful versus non-painful schwannomatosis schwannomas, much the way that was reported in NF2 schwannomas. Also, Kim Ostro has reported that the painful schwannomatosis schwannomas seem to have secretomes. They secrete different patterns of proteins compared to the non-painful schwannomas from schwannomatosis patients. And this is consistent with data that we've also reported at this meeting several years ago, showing that at least in SMARCB1 mutant schwannomas, there seems to be factors released that can influence pain signaling in the schwannomatosis background. So we've been looking at the SMARCB1 gene with regards to pain more carefully. And SMARCB1, as you recall, is part of the SWI/SNF chromatin remodeling complex that can regulate gene transcription. Mutations in SMARCB1 have largely been linked to rhabdoid tumors. However, more recently, because of the discovery of schwannomatosis, it's found there as well, where you see SMARCB1 loss in a significant number of familial and a smaller number of sporadic schwannomatosis cases. We generated a inducible conditional knockout of SMARCB1 in Schwann cells using the PLP-Cree ESR1 mouse from Brian Popko and an SNF5, another name for SMARCB1, Flox mouse from Charlie Roberts. And we've characterized these mice, and I've described these before. Steve Matsumoto also has described it at the meeting here before. But one of the most interesting phenotypes of these mice is that they have increased sensitivity to capsaicin. So if you inject capsaicin into the foot pad of the mouse and then measure the amount of time they lick their paw, the SMARCB1 mice always spend much more time licking their paw after the injection than the wild-type mice or the Flox mice that don't have Cree recombination. And so that suggests that, in fact, there's something going on with capsaicin sensitivity as a result of the loss of SMARCB1. And a receptor that responds to capsaicin is TRPV1. And so when we eat hot sauce or hot peppers, the capsaicin that we are tasting and reacting to is functioning via the TRPV1 receptor, giving us that burning sensation. When we looked at TRPV1 in our Schwann cell targeted mice, so these are mice that have, again, mutations in SMARCB1 only in the Schwann cells, the dorsal root ganglion neurons are changed. So what we look at is the TRPV1 expression. You can see here in the wild type animal, this level of TRPV1 in red. This is IB4, which labels the non-peptidergic neurons. And here's the merge. You can see they're not co-localizing very well there. And then if you look in the SMARCB1 mutants, there's higher levels of expression amongst neurons. And what's interesting is when you look at the IB4 expression co-localizing, now you see IB4 positive cells that are also TRPV1 positive, indicating that we have not only more neurons that are TRPV1 positive, but more non-peptinergic neurons that are positive. We can recapitulate the same phenotype in vitro by adding conditioned media from the SMARCB1 mutant Schwann cells to dorsal ganglion cultures. This is now the control, exchange for TRPV1 and IB4, and there's the merge. And now if you look at the SMARCB1 staining, TRPV1 staining in the SMARCB1 conditioned media treated cultures, you see much more expression, and again, it's overlapping with IB4, and this is quantified here. We see the same thing when we take human schwannomas that are SMARCB1 low or mutant in expression for SMARCB1. The conditioned media from these cells also induces TRPV1 expression in wild-type dorsal root ganglion neurons compared to human Schwann cell conditioned medium, which does not. Furthermore, as an indicator of activity, we've done cobalt uptake assays in the SMARCB1 treated dorsal root ganglion cultures and compared again to wild-type Schwann cell control conditioned medium. The SMARCB1 knockout conditioned medium dramatically increases cobalt uptake, and that's quantified here. So we're trying to understand the mechanism behind all of this, and we decided to do DNA microarray and proteomic analysis to find out what these Schwann cells are secreting when SMARCB1 is knocked out. And what we see is that in this assay, we can take a control virus here and an adenocre virus, add this then and let the cells incubate, take the conditioned media and take mRNA, and then do a DNA microanalysis and proteomic analysis to understand what's happening there. And we're using a click chemistry approach here to really look at the secretomes of these cultures. And what we find is that there are a number of changes that are occurring, but two kind of stood out. One is chemokine ligand 2, or CCL2, which has been implicated in pain in a number of studies. The receptors found in TRPV1 expressing nociceptive sensory neurons. If you apply it intrathecally, you wind up with thermal and mechanical hypersensitivity in rodents. TRPV1 receptor antagonists prevent this thermal hypersensitivity that can be induced by CCL2. And finally, it's been linked to increased TRPV1 transcription and activity. The other one that really stood out was IL-6. This can actually interestingly be induced by CCL2. The IL-6 and IL-6 receptor proteins are elevated in spinal cords and in the dorsal ganglia in a number of different pain models in rodents. Injection of IL-6 can cause mechanical allodynia or thermal hyperalgesia. And finally, intrathecal injection of an anti-IL-6 neutralizing antibody can alleviate pain-related behaviors. So here you can see the levels of induction. The scale is the same, but there's a significant increase in both CCL2 and a really dramatic increase in IL-6 secretion by the SMARCB1 mutant Schwann cells. Interestingly, we don't see the same changes in LZTR1 mutant Schwann cells, or knocked down Schwann cells, I should say. Fatima Benin in my lab was able to demonstrate that SMARCB1 actually interacts with the promoters of both IL-6 and CCL2. This is a chromatin immunoprecipitation assay up here. Here's the input, here's the IgG control, and you can see you can pull down the IL-6. SMARCB1 interacts with the IL-6 when you do the pull-down assays and with the CCL2 promoter. also Fatima generated luciferase constructs and was able to demonstrate that, in fact, if you add SNF5 to the cells, you actually can repress IL-6 transcription as well as CCL2 transcription, indicating that SMARCB1 is directly involved through this SWI/SNF complex, of course, with the transcriptional repression of both of these molecules. Finally, when we added IL-6 or CCL2 to dorsal ganglion cultures, and then looked for TRPV1 expression, comparing it to the control cultures that were untreated, both CCL2 and IL-6 were able to induce TRPV1 expression to a degree that was similar to that seen with conditioned media from SMARCB1 null Schwann cells. Cristina Fernandez-Valle analyzed these in schwannomatosis schwannomas for us, and in fact found that CCL2 and IL-6 are elevated in these tumors, as shown here. So these data suggest a model that involves SMARCB1 interacting with the IL-6 and CCL2 promoters. It probably is also regulating transcription directly of other genes that may be contributing to these painful phenotypes. And so when SMARCB1 is mutated, of course, what's going to happen is you're going to de-repress the transcription of IL-6 and CCL2, leading to this increase in IL-6 CCL2 secretion by affected Schwann cells. So the model we're working on then is that the SMARCB1 null Schwann cell is releasing these factors. These are being taken up by nearby dorsal ganglion neuron axons, leading to DRG, that neurons that have elevated TRPV1 expression. I should note we also see elevated TRPA1 and CGRP in our model, and this leads to this increased pain sensitivity. So the question is, if you block the synthesis or release of these factors or their activity, could you then prevent all of this from happening? Or could you block TRPV1 expression or activity? There are antagonists to TRPV1 that have been looked at. They've all had some serious side effects, so it's not clear if they're going to be used in the clinic at any time soon. So we are really pursuing the possibility of using factors that block this kind of change here to prevent the chemokine and cytokine synthesis by the Schwann cells. So in conclusion, pain is a common symptom in NF1, NF2, and schwannomatosis patients. NF1, NF2, and schwannomatosis patients appear to experience different types of pain, though. And what's more, there seem to be mechanisms of pain sensitivity that may be altered as a result of specific mechanisms related to the types of mutations in the different genes that occur in these diseases. So finally, pain in schwannomatosis patients with SMARCB1 mutations may be related to the release of specific chemokines and cytokines that influence pain signaling in sensory neurons, affording us a possibility for intervention. And our data also suggests that the SMARCB1 and LZTR1 mutations in schwannomatosis patients might lead to different types of pain issues. So thank you for paying attention to the talk, and I'm happy to take any questions.
Okay, thank you, Dr. Sherman. That was great. We actually don't have any questions from the audience right now. So if there are any questions, the audience can still go ahead and enter them, and I can call you back up if that happens. But I think in the meantime, I will go ahead and introduce our next speaker, Dr. Justin Jordan, who is an assistant professor at Harvard Medical School, who practices at Massachusetts General Hospital in the Family Center for Neurofibromatosis and the Pappas Center for Neuro-Oncology. And his top focus is on genotype and whole body MRI metrics in schwannomatosis. Dr. Jordan.
Great, thank you very much. I'd like to say just a huge thanks for inviting me to present today at the meeting. So my name is Justin Jordan. I'm the director for the Family Center for Neurofibromatosis at Mass General Hospital, and I'm excited to talk to you today about genotype and pain and whole body MRI in patients with schwannomatosis. So you'll see my disclosures here. None of them are directly related to schwannomatosis or to the work that I'm describing today. So by way of background, I don't think there's any need to belabor to this audience, especially after the great talks we've just heard, the important relationships between schwannomatosis and pain. I will just say that chronic pain is a dominant feature of schwannomatosis, as we've heard. Pain is often unrelated to specific tumors or to sites of tumors, as has been described previously. And the majority of patients with schwannomatosis do take chronic pain medications, as has been reported previously. The genetics of schwannomatosis are not yet fully understood. To date, we've identified that SMARCB1 and LZTR1 are associated with schwannomatosis, but in reality, that leaves the majority of patients with this disease in neither of those categories. And you can see that the differential genetic findings between familial patients who are SMARCB1 dominant and sporadic patients who are LZTR1 dominant still leaves a lot of unknown genetics in these patients, which is an important consideration when undergoing any sort of genotype-phenotype correlation analysis. And while we don't have a strong relationship between schwannoma locations to date, we do know a little bit about, between schwannoma locations and pain, we do know a little bit about where schwannomas happen in patients with schwannomatosis. So the vast majority of patients do have at least one peripheral nerve schwannoma, which is mostly in the extremities. About 3/4 of patients have spinal schwannomas, and you'll see that 20 to 30% of patients have subcutaneous schwannomas, while a much smaller number have non-vestibular intracranial tumors. So I'll tell you about the work that we have done so far, which is ongoing. So the underpinning of this analysis is really related to whole body MRI, which, as most of you know in the audience, is an incredibly important field in neurofibromatosis to help us better understand the total body burden of tumors in these very variable diseases. So in this study, we used a 1.5 Tesla machine with a whole body coil. We took coronal images only with 5mm cuts, largely related to time, trying to optimize the value and resolution of images, but not keep patients in the scanner for eons. And we did not use gadolinium contrast for these studies. Instead, we relied heavily on STIR images to help us identify tumors. We had a board-certified radiologist identify the tumors, and then with some wonderful post-processing, use computerized 3D tumor volumetry to determine not only the total number of tumors, but also the total burden of tumors and individual tumor sizes. We've also developed a sequencing pipeline that we applied to the patients I'm describing today. The patients have previously consented to leaving blood with the clinic to develop a lymphoblast cell line. And from those lymphoblasts, we extracted DNA here and applied a custom biotinylated capture system, which captures the entire SMARCB1, LZTR1, and NF2 genes. The NF2 gene, of course, is in recognition of the enormous phenotypic overlap between NF2 and schwannomatosis to ensure that no patient is misdiagnosed in our cohort. And then for every patient in this study, we've looked at pain by a couple means. The first is the VAS, which simply but importantly is a horizontal line that's 10 centimeters in length, and patients are asked to make a mark at the location of their current pain from no pain on the left to pain is as bad as it could possibly be on the right. And then we also collected the patient-reported SF36 questionnaire, which looks at a number of quality of life metrics, including bodily pain, which we'll talk about here today. So from this, we began with a cohort of 51 patients who had both schwannomatosis and a whole body MRI available with analysis having been performed. 14 of those individuals either did not consent to leaving germline DNA for research or had no available genetic information already for our analyses. And so that left us with 37 patients that I'll describe today. 30 of them we performed our gene capture and sequencing pipeline on, and 7 of them had previously available sequencing results with a mutation identified related to their schwannomatosis. So on the technical side, the pipeline worked very well. The median coverage at each of the loci was 1000x. With a 72% efficiency of capture within the target region. So the assay worked very well with these biotinylated capture beads. No deleterious NF2 mutations were identified in the germline of these individuals, importantly. And here you see a table of the variants that we didn't identify. And so there were 5 LZTR1 variants, and 8 SMARCB1 variants included here. I'll draw your attention to just a couple things. The first is there are superscript A and B next to several of the SMARCB1 mutation variants, and those are familial. So the Bs were a family, the As were a family. And then secondly, there are a number of variants listed here that have asterisks next to them, and each of those has been previously described by Dr. Smith in prior publication. All of the variants that we identified here were confirmed with Sanger sequencing, and for those for which functional change was not previously defined, some in silico analysis was performed for those. So we had a number of patient demographic and characteristics that we collected on the various patients in this study, and we looked across the different groups. I'm going to highlight for you some of the important ones here. I'll orient you to the table. So each of those characteristics that we evaluated is here on the left, and the columns are the 5 patients in this group with LZTR1 pathogenic variants, the 15 patients with SMARCB1 pathogenic variants, 17 patients in whom we found neither, and then we looked at the statistics across all 3 groups and across only the patients with LZTR1 and SMARCB1. And the rationale for that is we presume that patients who have neither LZTR1 nor SMARCB1 mutations may be a heterogeneous population, and so may not make complete sense to include as a third group per se. So the things that stuck out specifically here, number one, age at diagnosis was no different between these groups, which I think is meaningful. I will highlight specifically that the pain score, the median pain score, was significantly higher in patients with LZTR1 than in patients with SMARCB1. And so you can see the numbers were 3.9 median versus 0.5 median. Relatedly, we also identified that the median bodily pain SF36 subscore was significantly worse, which is a lower score in that measure, compared to SMARCB1. And actually, across all 3 groups, SMARCB1 tended to be the mildest bodily pain SF36 subscore compared to LZTR1 or other. And then the last thing I'll point out specifically here is, well, a couple other things. There was no difference in number of tumors or total body tumor volume between the genetic groups, but we did find specifically that spinal schwannomas were much more prevalent in the LZTR1 patients, and 100% of them, relative to SMARCB1 or the other category, which were each about 40%. We looked at the correlation between tumor burden and pain across all patients. And you can see that while there was no correlation between the VAS pain level and tumor number, we did find a relationship between the VAS pain level and the total body tumor volume. And this is actually confirmatory of previous work published by Vanessa Merker in our group in 2012. There was no difference in pain or in pain-related quality of life depending on the presence of spinal schwannomas. So that significant difference between those two groups didn't relate specifically to pain across tumor burden. We looked within each of the individual mutational groups in the LZTR1 and the SMARCB1 categories, and there was no significant correlation between pain and tumor number or pain and total body tumor volume in those groups. So in conclusion, we identified here that pain does correlate with the total body tumor volume. And as I noted, this does confirm previous work published by Vanessa Merker in our group. We saw that pain and pain-related quality of life are significantly worse in patients with LZTR1 mutation than SMARCB1 mutations. And it's really interesting to step back and think about the 3-step 4-hit hypothesis of tumor generation with schwannomatosis, wherein we believe that both copies of NF2 and either LZTR1 or SMARCB1 must be hit before a tumor develops, but perhaps based on this data, the underlying germline mutation alone may be sufficient to relate to the pain phenotype that is so prevalent among these patients. We saw that more spinal schwannomas were present in the LZTR1 mutant patients than in other groups. That did not relate to pain. And while I think that the tumor location information is important, we also can relate back to previous publications that the tumor location is not a particular correlation with pain. So this confirms those prior findings. And lastly, there was no significant difference between tumor burden in LZTR1 patients or SMARCB1 patients, which I think is a meaningful finding as we address these patients in clinic and try to understand prognosis, for instance. There's a number of future directions for this work. Number one, a longitudinal multi-institutional pain survey is underway, and I would encourage you to tune in at 1:45 today to hear Ms. Ashkenazi report on work related to that pain survey. There are new schwannomatosis-specific pain clinical trials. I would encourage you to tune in later to hear Ms. Da present on one of those studies at 3 p.m. today. And there are a couple other concept trials that have been discussed, including both in the United States and in Europe, which I hope will be informed in large part by the genetic information that we're talking about here. And then lastly, I think to better understand and plan for future trials, a natural history of schwannomatosis certainly needs to be better understood as we dig further and further into genotype and relating those genotype-phenotype correlations. I'd like to acknowledge the enormous number of people who helped with this work. Specifically, Scott Plotkin has just been a tremendous mentor in this and all other things that I've worked on. Miriam Smith has been a terrific collaborator from Manchester on this and other work, and many people you can see here, too many to name, but have been truly remarkable for all of their help. So thank you very much, and I'm happy to take any questions.
Thanks so much, Dr. Jordan. We do have several questions for you. We also have a question for Dr. Sherman that I'll call up in a few minutes. So Dr. Sherman, you're on standby. Dr. Jordan, one question is, how much can the understanding of pain chemokines from schwannomatosis be translated to understanding PN-induced pain in NF1?
It's a fascinating question, and actually Dr. Sherman and I were just talking about this earlier today. The talk yesterday on LZTR1 biology and the relationship back to RAS really, I think, raised a lot of interesting hypotheses for a commonality in pain generation between schwannomatosis and NF1. And so I think that there's a lot more to come in understanding the probably subtle differences, but potential commonalities and how to address those in terms of clinical trial planning.
Okay. Another question. Do inflammatory cytokines like IL-6 induce nociceptive or neuropathic pain, or both?
It's a great question. Dr. Sherman, maybe that's best addressed for him. I can't say I can speak to that with expertise.
Okay, well, we will save that for Dr. Sherman. So one more for you, Dr. Jordan. And I know this may be something that you touched on somewhat in your talk, but there are a few studies that show that the LZTR1 negative SWNTS patients are the ones that experience the most pain. Can you speculate on the LZTR1 mechanisms?
Yeah, it's a great point. And I think in reality, there's a lot more basic science to be done to try to understand whether there is a relationship to CRMP2 and the various other pathways like we see in NF1, whether there may be commonalities there.
Great. Thank you so much, Dr. Jordan.
Thank you.
Great talk. So Dr. Sherman is back with us as well. And a question for you that I just posed was about whether inflammatory cytokines like IL-6 induce nociceptive or neuropathic pain, or both. Do you have a comment on that?
I mean, it can be sort of a both type of situation, I think. I mean, it's a neuropathic mechanism in a way because it's something that's changed in the nervous system, obviously, in this case, but also it's part of an inflammatory cascade. So, and that could be in response to an injury like a mechanical injury or even thermal or chemical injury. So it can actually cause similar types of responses depending on the context.
Okay.
And then I actually had a question for Justin. If you're still there, Justin, do patients with SMARCB1 mutations still have pain that is significant enough to affect their quality of life? I remember when I read your paper, it's like there was a comparison. It's less maybe in that particular cohort at least. But do they still have episodes at least of painful situations where their quality of life is still significantly affected? And does their pain differ in quality from the LZTR1 patient's pain?
That's great questions, both of them. First, I'll just sort of point back to the talk later today for our longitudinal pain survey, which I think will be informative for that. Certainly, there was pain in this group, and as you pointed out, this is a small group. So my sense is that SMARCB1 mutant schwannomatosis is also a painful disease. Potentially, there's a difference in the amount of pain and the pain relationship to quality of life between LZTR1 and SMARCB1, but I absolutely think that it's still an important focus for ongoing research in that group.
Okay, great.
Dr. Sherman, one last question for you. Someone from the audience asked, were the schwannomas in Cristina Fernandez-Valle's experiment all SMARCB1 mutated?
The one that we looked at and that I showed demonstrated loss of SMARCB1 expression. So it was probably one of those mutations where there's low SMARCB1. And then the ones that we cultured, we went back and verified the same thing. So it was they were very low in their SMARCB1 levels compared to the wild-type Schwann cells that we used. We're also validating that experiment in human Schwann cells where we're knocking down SMARCB1 in wild-type human Schwann cells. So.
Okay. Great. Well, thanks for coming back to answer those questions, Dr. Sherman, and thank you, Dr. Jordan, again for your talk. We will move on to our final talk of the session, which is by Dr. Frank Buono. He is an associate research scientist in the Department of Psychiatry at the Yale School of Medicine, and his talk will be on the qualitative understanding of pain interests across medical experts and individuals suffering from NF1.
Good morning, everyone. My name is Frank Buono. I'm an associate research scientist in the Department of Psychiatry at Yale School of Medicine. and I'm happy to present my current talk, which is a qualitative understanding of pain interest across medical experts and individuals with NF1. I do have no disclosures to report at this time. As we all pretty much know, neurofibromatosis type 1 is an autosomal genetic disorder affecting about 1 in 3,000 individuals worldwide. And the recent research study has shown that about 50% of individuals with NF1, specifically adults, are reporting significant pain and discomfort across multiple pain indices. Being that so, mobile health applications can only increase the pain management resources for individuals at a lower cost than other conventional treatments, and will affect a greater amount of individuals as far as in real world settings and whatnot. But once established, it can also be tailored to specific illnesses or conditions. The I Can Cope mobile application, which is coming out of Dr. Jen Stinson's lab at University of Toronto, is an innovative mobile application which was initially developed to help to address pain self-management needs for adolescents and young adults with chronic pain, utilizing a CBT and mindfulness-based approach for individuals. The ICC application has been expanded to juvenile idiopathic arthritis, sickle cell disease, and postoperative pain. For the current study, what we would like to do, what we did was we proposed to adapt the ICC platform to the specific needs of adults with NF1. And following the American Psychological Association and American Educational Research Association standards for education and psychological testing, we recruited both experts in the field of who are treating the individuals with NF1 and individuals who are dealing with the chronic pain themselves to see their perceived acceptability and the barriers and facilitators to use of this mobile application. A qualitative study was done, and we had 32 individuals with NF1 who successfully completed the study, and we had 10 individuals who were clinicians. Recruitment came from individuals from the NF1 group through either a Facebook blast or an email on 2 different NF advocacy group sites. Recruitment for expert came from a website posting on the U.S. Association for Study of Pain and or direct conversation with NF Network clinician page through medical doctors. The study was approved by the Institutional Review Board at Yale School of Medicine. To look at the patients and expert breakdown, we saw a preponderance of females within the NF patient group as 25 of the 32. And looking at the pain that they were reporting at baseline for this group, we saw that most of them were in moderate levels of pain, registering at 4.8 and 4.9 respectively for severity and interference when looking at the brief pain inventory on a scale of 0 to 10. And looking at the professional training, we noticed that we had 2 pain psychologists, 5 neurologists, 2 primary care physicians, and 1 neuro-oncologist, showing also most of the professionals were seasoned and that they were having an average of 19.2 years of experience, and the range between 7 and 38. So each focus group lasted about an hour in length. We tried to keep the groups relatively small. For the NF group, it was anywhere from 6 to 8 individuals, and the clinicians were around 4 individuals per grouping. All individuals were given a secure and password-enforced video conference link, and 2 members of the team were there to facilitate each session. The 4 domains that we focused on were the perceived barriers and facilitators for using the mobile application to manage pain, the perceived acceptability and benefits of the proposed application, context in which the application would most likely be used, and the recommendations for possible additions or modifications of the application. In looking at the results, we found 6 themes across 2 different levels, these 2 levels being the individual level and the app level. And of those 6 themes, we have lifestyle, reasons for using, concerns, desired content, desired features, and format consideration. In looking at these 6 different themes, we noticed that there was a higher likelihood, if we manage these appropriately, to get a higher motivation of use and reuse of multiplication. For the sake of time, I'm going to just focus on a couple different content areas and selected quotes. One of them was from the expert group within desired content, and we had most of the experts agreed that there is a plethora of research on pain education and the importance of pain education within pain management strategy. However, there is little to their knowledge about how pain education affects NF1, and they thought it would be very important, if not seminal, to include within the multiplication. And based on this feedback, we included a subsection of pain education and why it's critically important for individuals with NF1 in the actual clinical trial mobile application. One area of intent for individuals with NF was to look at a community function feature. This feature was echoed by 31 of the 32 individuals, saying that this would be a very critical portion of the mobile application, because we had a large set of individuals who had never actually seen another individual with NF before, or they lived in a rural setting and didn't have a lot of exposure to good standard level care. And they said that they would like to have other NF individuals around so they could talk to and gain hope and cope from this mobile application. Lastly, we also noticed that within format considerations, that most of the NF1 individuals wanted the app to be simple and fun. They didn't want something which was extraneous. They didn't want something to be heavily doctor-oriented. They wanted to make sure that it was easy for them to use and to check in very quickly. To summarize really quickly, both groups were excited about the potential to have an ICC mobile application for NF1, so that we could look at pain and how pain affects their daily lives. Both providers and patients noted that the importance of tracking pain intensity, duration, quality, and the directionality were critical, and that patients also should know the ease of use, which would be very important for their daily use and daily motivation of utilization. One area that I'd like to focus on, which there was a different opinion on, was how the groups thought of looking at the pain data and what would be most important to them. The NF group appeared to be interested in gathering the nuanced and detailed information as much as possible to have. So they wanted to know if the pain was, a tingling, a numbing sensation. They wanted to know what time the pain had started, if it was multiple areas, if it was a singular area, whereas the pain experts were more focused on the possible associations between pain, mood, the nutrition, and the activity, or the lack of activity. And to our knowledge, this is the first current study that has been looked at within both individuals with NF1 and the pain experts. With that, I'd like to thank my funding through the CDMRP. I want to also thank my team members at Yale School of Medicine, Connecticut Children's Hospital, and the Hospital for Sick Children. I can give my references if you need to see. And I'd like to thank you for the time, and I'm happy to answer any questions or thoughts. Have a good rest of the day.
Thank you so much, Dr. Buono. I have a question, which is, it seems to me that there's a lot of potential for this app to be modified and made really helpful, not only for people with NF1, but potentially people with NF2 and schwannomatosis. Do you have any thoughts about that?
Yeah, that's actually the future this idea of what we want to do. I think that the idea of working with NF2 and schwannomatosis is clearly another next step. We're currently in the clinical trial as we speak for the NF1 and should have some good preliminary data in the next month or two, and hoping to find some funding sources for look at NF2 and schwannomatosis.
Okay, great. And then we have a question from an audience member. How do you think we should balance the needs of patients and experts in drug research and clinical practice?
It's a fantastic question, and I think that my colleagues on this call could probably provide a better explanation than I could. But I think I can give you a patient perspective because I actually suffer from NF. That it is a critical piece for the individuals to have their voice heard, and I think that if the most that you can provide to them so that they can understand is very critical. But I think that Dr. Jordan or that Larry could probably even provide some better clarification for that too.
Yeah, I'm happy to hear from either of them. If either of them has a comment on that, they can turn their cameras on and add to that discussion. But I definitely think, you did a nice job of kind of communicating, here's where the experts were focused and here's where the patients were focused. And it's not that they were at odds with each other. It's just that they have different kind of areas that they really honed in on.
And yeah, I think that's pretty clear, at least, from what we saw is that, patients want to know this granularity and really understand why the behaviors are occurring, at certain times, rather than what I was saying with the clinicians. We're just looking at the associations, and I think that is something which is interesting to me because suffering from NF, I'm not really interested in much as the pain, the numbness, the tingling, but really looking at how long it occurs for, what is affecting it.
Yeah, and the things that you mentioned the experts were particularly interested in were things like how does pain relate to nutrition and activity level, really kind of displaying a holistic approach to consideration of pain. Dr. Jordan, do you have any comments that you'd like to add?
Yeah, I would just highlight, Frank, that was a wonderful talk. I would highlight that this really underlines the importance of involving patients in every bit of research planning to make sure that whatever it is that we're discovering, improving upon is ultimately important to those affected by neurofibromatosis.
Yeah, that's great. Well, that was an excellent talk, Dr. Buono. Thank you so much. And I want to thank all of our speakers today for some really informative presentations. We are ending about 2 or 3 minutes early, and then we have a 5-minute break. So I invite everyone to take a break, stretch your legs, do whatever you need to do. I think we're back at around 10:50 this morning. Thank you, everyone, for attending this session.